Custom microscope design for 3D optical imaging. In modern science and medicine, 3D microscopic imaging is essential for exploring the complex architecture of biological tissues and nanoscale materials. However, achieving high-resolution 3D images presents challenges including limitations in optical techniques and data processing. Overcoming these hurdles is vital for advancing innovations in medicine and material science. 3D microscopic imaging faces significant challenges that hinder its effectiveness. Axial scanning methods extend scan times, reducing temporal resolution and complicating specimen stabilization. Using multiple detectors can lower measurement quality and complicate hardware design, impacting data accuracy and analysis of dynamic processes. Additionally, large datasets strain storage and management, while detector limitations affect imaging resolution and fidelity. Innovative solutions in instrumentation and computation and data management are essential to improve 3D imaging in science and medicine. This gives us overall approach of our project. The proposed framework comprises three key components arranged sequentially to address the inherent challenges. Firstly, at the forefront of the framework lies the forward imaging model designed to capture depth resolution within the microscopic specimens. This component serves as the foundation, ensuring the faithful representation of 3D structures onto planar detectors. Following this, we have the detector model designed with practical limitations. Finally, a reconstruction framework for leveraging the encoded information from planar measurements to reconstruct the 3D specimens. The forward model is essential for converting a 3D specimen into a 2D detector image. It employs a vector-based point spread function model to simulate the imaging system's behavior accurately. This process begins with a laser light source illuminating the sample. The light then passes through a digital micromirror device, which consists of tiny mirrors that create structured light patterns. These patterns allow us to capture detailed information from the 3D object. By changing the DMD patterns, we effectively take multiple snapshots from various angles or depths. Despite working with a 3D object, the detector captures only a 2D image from the middle plane at each step. Therefore, a series of measurements with different DMD patterns is necessary to gather sufficient information for reconstructing the entire 3D structure. Each 2D image provides new data, and the structure illumination encodes depth information capturing details from different planes. The forward model also considers detector limitations such as lower resolution and noise, ensuring a comprehensive depiction of the imaging process. By combining these multiple 2D images, we can reconstruct a detailed 3D model at the desired resolution. This approach is crucial for efficiently imaging complex structures, overcoming the limitations of single measurements, and ensuring we have enough data to build high-resolution 3D images. The reconstruction problem entails estimating the original 3D specimen from a series of measurements with the knowledge about the forward process. While the imaging simulation occurs at a finer sample level, reconstructing the specimen at such a high resolution poses challenges due to physical limitations. So we set our target as a low resolution object formed by 3D average pooling of the analog volume. Additionally, the forward process is approximated to match the digital volume via the proposed model approximation strategy. So under this method, we used plug and play. We combined three two-dimensional denoisers to implement a highly effective three-dimensional prior model. Our approach integrates three two-dimensional denoising convolutional neural network priors using multi-agent consensus equilibrium. MACE is an extension of the PNP framework that formulates the inversion problem using an equilibrium equation in contrast to optimization and allows for the use of multiple prior models and agents. These are the qualitative results from our system. We imaged blood vessels in the brain known as cortical vasculature. We also imaged the neural cells. These are the quantitative results. In conclusion, our custom microscope design leverages structured illumination and advanced image reconstruction to efficiently enhance 3D imaging.